Greetings. I hope you guys are okay. Welcome to SBCA021 revision session. So let's look into part one of SBCA021, which is basically biochemistry. So it speaks a lot about the central dogma. So we have our first question here. 3.1 says discuss the three differences between DNA and RNA. So this is one of the questions that they are likely to come. So when you discuss their differences, you can just start by tabulating between the two. So speak about DNA this side, speak about RNA this side. So the first that you want to use for differentiating, you can speak about the sugars. So we know that the sugars in DNA is deoxy, ribose, sugar, and the sugar in RNA is what? Is ribose, sugar. And then the you can also further talk about the nitrogenous bases. So the nitrogenous base that is present in DNA and is not present in RNA, that is what? That is thiamine. And the nitrogenous base that is only present in RNA is what? Is uracil. And then you can also talk about the strand. DNA is double stranded, and RNA is what? Is single stranded. So you can also add more and talk about the stability. You know that DNA is more stable than RNA. So more stable, less stable. So you can also put these differences into words wherein you speak about, you discuss how the sugar in a DNA differs from that one in RNA, and also discuss in ways that okay, in DNA we have nitrogenous bases such as guanine, cytosine, and adenine, and in place of instead of uracil we have thiamine, and then in RNA instead of thiamine we have uracil, and then you go on and speak about the strengths, and you go on and speak about the stability. So moving on to the second question, 3.2, which speaks about, it just says name the bond that binds nucleotides to form nucleic acid. Here we're talking about the bonds that joins the nucleotides that you find in RNA or in DNA. So that is the phosphodiester bond. So 3.2, 3.3, you have it says name the bond that binds the nitrogenous bases in the nucleic acid. We're talking about, when you speak about nitrogenous bases, we're talking about adenine, guanine, cytosine, thiamine, those ones. So the bonds in between the guanine and cytosine and the bond in between adenine and thiamine is what we call them the hydrogen bonds. So moving on to 3.4, they say, okay, here we have to explain. So, and it has got 4 marks there. This means we must have about 4 points when we explain. So they say, why would a DNA sequence with a higher guanine cytosine content have a higher melting temperature compared to DNA sequences with a higher adenine thiamine content? This is basically because of the number of hydrogen bonds that are between the nitrogenous bases. So our first point, we can talk about that, wherein we will say that the guanine cytosine base pairs They are held by what? By three hydrogen bonds. Hydrogen bonds, in a sense that you have something of this kind. One, two, three. See. So while the bonds, the A T base pairs, they have only two hydrogen bonds. So this tells you about the amount of energy in terms of heat that would be required to break the bonds between the guanine and cytosine compared to the energy that would be required to break the bonds between the adenine and thymine. So another point we can speak about what I just said now. So we know, okay, uh, more hydrogen bonds, they mean what? They mean they mean more energy. Where is it up? Would be required, right? More energy. When you talk about energy, we're speaking about energy in terms of what? Of heat, right? It's gonna be required to do what? To break the bonds, the bonds between the what? The the nitrogenous bases. So in this case, it's gonna be required to break the what? The DNA double strand, right? So remember, we're still trying to answer the question as whether why, right? So this tells us that, okay, uh, a higher a higher guanine cytosine content means more hydrogen bonds in the molecule, right? Which molecule? The DNA strand or the double strand, right? So furthermore, on our last point, this makes DNA to be what? To be more thermally stable. We said that DNA is more stable up there when differentiating it from what? From RNA. So this makes DNA more thermally stable. When we talk about thermally stable, I mean in terms of heat. So this will result in high melting temperature. In temperature, in terms of the temperature Tm, which I'm speaking about there. So this is the reason why the DNA sequences with high GC content have a high melting temperature compared to DNA sequences with a high AT content. So that's basically the answer to the question. So now let's look at 3.5. So in 3.5, we're speaking about true or false. So you have 3.5 0.1, you have 3.5.2, you have 3.5.3, you have 3.5.4, you have 3.5.5. So the first one says DNA repair pathways can correct damage caused by environmental factors such as UV radiation and chemical exposure. This is true. This is true. And then what about 3.5.2? Mismatch repair is a DNA repair pathway that corrects errors during DNA replication, which is also true because it deals with the mismatching. If adenine is not paired to its proper nitrogenous base, then that mismatch repair will facilitate the repairing of DNA. So this is true. And then 3.5.3, it says cells cannot survive without DNA repair mechanism. Yes, of course, they cannot survive without it because this means there will be damage to those cells. So if there is damage, how are they going to survive? They are not going to survive. So this is what also true. And then 3.5.4, DNA repair mechanisms are highly accurate and never introduce errors during repair process. This is false. They do introduce errors at some point, but those errors they also get fixed, even though it's not always, but they do introduce errors. So this is false. And then both prokaryotic and eukaryotic cells, they have the same DNA repair pathways. This is also false. Reason being, they do have the same mechanism. The mechanism is the same, but the pathways they differ in terms of complexity and also in terms of the proteins that are involved. So they cannot be the same. So that is false. Now let's look at question 4. We have our structure there. We have 4.1, 4.1, we have 4.1.1, we have 4.1.2, we have 4.1.3. So 4.1.1, it says name the parts or components represented by A, B, and C. Okay, we have A. Let me get this and do this. We have B, we have C. Now, on that day, we see SER, and we can tell that this is a transfer RNA. So a transfer RNA, what does it do? It carries an amino acid. So what is being carried there on A is what? Is an amino acid. And then B, or B is what? Oh, that tells us that now we have an anticodon there because it is carrying the what? We know that at the base of the transfer RNA we have what? We have anticodons there. So that is anticodon. And then what about C? C, that is the mRNA, of course. So mRNA. That's it. Moving to 4.1.2, give the sequence of the component represented by B on a diagram above. The component represented by what? By B. They want the sequence. So we know that the sequence is what? Is basically the opposite of what we see on the what? On the mRNA. So they must complement each other. So this means A will be what in the anticodon will be U. And then this means G will be what? Will be C. And U will be what? Will be A. But here is a catch. You also have to what? To indicate the direction. The direction of the anticodon strength, or the direction of anticodon and the direction of mRNA, they are different. If the direction of mRNA is 5' to 3', this means the direction of the anticodon strand there was going to be 3' to 5'. So this is how you're going to write it. You're going to write 3' U C A to 5'. So that is the sequence of the component represented by what? pi b on the diagram above. So let's look at 4.1.3. So it says name and define the process that result in the synthesis of the component represented by label C. So that is how is mRNA formed? It is formed through what? Through transcription. That is transcription. Now the question is not oh, they say name and define the process. So how do we define it? This is a process. Is a process whereby an enzyme RNA polymerase uses the what? It uses a DNA template to do it, to synthesize a complementary single-stranded mRNA molecule. You can also mention the direction that in 5' to 3' direction. So that is it. So we also have 4.2. So in 4.2, they say transcribe and translate the DNA sequence 5' 5' ATG TAC and that. So and they say you must show all your workings there. So for this one, you can use you can do it anyhow you prefer, but I think maybe let's do it this way. Let's first start with transcription. We know transcription is the conversion from DNA to mRNA, right? So this means that we have you have DNA, and remember your strand must have the direction. Whatever that you write there, the sequence must have a direction. The sequence is saying 5' to 3'. So this means that even your sequence of mRNA must have a direction. So in this case, it says what? 5' to 3'. So let's tabulate this and do like this, and have the mRNA this side. This is gonna be in the same direction, 5' to 3'. This is just a way of making it much easier, but you don't have to stick to the way I'm doing it. But I saw it as an easiest method to apply. So you have ATG, you have TAC, which is this is coming from the same strand that you are given. You have GCA, you have TGG, you have CTA, you have TAA. So what's gonna match what is above in an mRNA? So this is what you're gonna have for A. A will bind with what? With your uracil. So you're gonna have U, and then T with what? With A, we're gonna have UAC. And this side we're gonna have A UG. This side you're gonna have CGU. This side you're gonna have ACC. This side you're gonna have what? GAU. And then this side you're gonna have what? AUU. Remember to change what? To replace the thiamine of DNA with the what? With the uracil in mRNA strand. So now moving forward, this means our mRNA strand is gonna be what? It's just gonna be 5 prime what A U. Okay, you'll forgive me with the background noise there. U A C, and then A U G C G U A C C G A U, and then A U U. Your spacings must be proper there. Don't so, and then at the end you must have 3 prime there. So this is your mRNA strand. So now we take the mRNA to what? To the translation. This is what we translate. So in translation, you have what? You have the mRNA codon, and also you must have a corresponding amino acid, right? And then this side you have what? The mRNA codon. So what happens from there? Our codons, they are what? Remember, you have what? U A C. You have A U G. You have C G U. You have A C C. You have G A U. You have A U U. So what is the corresponding amino acid for each codon that you see here? The corresponding amino acid, how are you gonna check it? You go to the what? To your codon chart. So the first base, we have UAC. The first base is what? Is U here. First base is U. And then the second base is gonna be what? The second base is A here. And then what is the third base? The third base is what? Is C. So we check here on C. So it's giving us what? It's giving us what? ACU here. What is the amino acid? The amino acid is what? It's tyrosine. Which you can also write TYR, but I'll advise that you write them in full. And then what about AUG? You do AUG as well. And then when you do AUG, the first base is what? It's A, and then it's gonna be A, U, and then G is giving you what? Methionine. Methionine. And then CGU as well. I'm sorry about that. You have CGU, which gives you arginine. And then you also have a CC, which gives you threonine. I think it's E, threonine. And then you also have GAU, which gives you aspartic acid. And then AUU finally, which gives you isoleucine. Know how to spell this amino acids. So this means your polypeptide chain is gonna be what? It's gonna be what? It's gonna be TYR bonded to what? To met, bonded to what? To arginine, represented by ARG, and then 309, and then what? Aspartic acid, and then isoleucine. So this tells you we don't have a stop codon here. So this tells you that the translation will occur again and again until it forms a stop codon. So that's it for those examples, and then we're gonna keep on continuing on making other examples and trying to see how we can attempt them. Thank you.